All right, HL students, this one's going to be amazing. So we're talking about C1.2, which is cellular respiration, and we need to be able to describe in a lot of detail how glucose is used to make ATP in mitochondria. So we're looking at the concept here of interaction and interdependence, and we're looking at page 370 to 383. The upside is once we learn this, photosynthesis seems quite straightforward. All right, here we go. So firstly, we need to understand that oxidation and reduction and what those things are. And so if one molecule is reduced, that means the other is oxidized. And we're going to be talking with photosynthesis here about NAD+, which is a reusable electron carrier that picks up 2 electrons and a hydrogen ion. So it turns into NADH or NADH. This process is called reduction. So where it goes from NAD+ to NADH, this is a reduction reaction. And if it were to lose hydrogen ions or to lose electrons, then it would be oxidation, so the other direction. So you can remember this from the acronym OIL-RIG, which is oxidation is loss and reduction is gain. Some bacteria oxidize hydrocarbons by adding oxygen. And that would also be an oxidation reaction, adding oxygen. Adding oxygen is considered oxidation because the oxygen pulls electrons to that part of the molecule, essentially pulling it away from other parts of the molecule. And so as a result, oxidation is lost. Those electrons have been lost from certain areas of a molecule. So losing oxygen then would be considered reduction. But we're mostly going to be looking at oxidation and reduction with regards to hydrogen ions and electrons. All right, so the cellular respiration in mitochondria is happening in 3 separate reaction processes. So the first one is in glycolysis, and this is actually happening in the cytoplasm of the cell. So this is outside of the mitochondria. Glucose is phosphorylated, and that's going to make it unstable. So that means ATP is added, adds phosphate groups to the glucose molecule, and it is eventually going to break apart because it becomes unstable. So it produces pyruvate. And the net gain on glycolysis is just 2 ATPs, and also it will make some NADHs. And those NADHs, the NADHs, are going to go into the mitochondria, and they're going to be used in the electron transport chain. So not only do we want ATP, but we want to make these electron carriers reduced so that they can be used in the electron transport chain. This is where most of the ATP is made. So glycolysis, the link reaction, and the Krebs cycle are all making these NADHs, those are the reduced NAD+, for usage in the electron transport chain. And the electron transport chain is going to use them in the intermembrane space. So that's going to be this space in here. And so when we're looking at a picture of a mitochondria here, we do need to know some of these components. So they have an outer membrane and an inner membrane, and then the space between the outer and inner membrane is the intermembrane space. And these cristae here are invigulations, like so per I can't remember the word I'm trying to say here, but bendiness of the membrane. So that's going to increase surface area to volume ratio. And then the inside of the cristae are called the matrix. And of course, inside here, there are going to be ribosomes, and there's going to be segments of mitochondrial DNA. If oxygen is missing in this process for glycolysis, pyruvate can be transferred to lactate and oxidize the NADH to remake NAD+, and that will allow this reaction to keep going to make more ATPs. But that will only produce a very small amount of ATP, and that's why you run out of energy if you're not getting enough oxygen. So we do need to know, unfortunately, glycolysis in quite a lot of detail. And so here we're looking at a glucose molecule. ATP is added to that glucose molecule, phosphorylating it. And so that's going to make it unstable. And then even more ATP is added to that glucose molecule, making it fructose 1,6-bisphosphate. So you don't need to name all of these things, but you will need to know glucose 6-phosphate. You will need to know fructose 1,6-phosphate. And then what is going to happen is this fructose 1,6-bisphosphate is going to restructure. And in that restructuring process, it's going to become G3P, glyceraldehyde 3-phosphate. So you can see here, this molecule has a phosphate group on it. And so then NAD plus a phosphate group that's just chilling in the cytoplasm there is going to be added onto that G3P. And in the process of the phosphate being added, the NAD plus is going to be reduced and it's going to make NADH. And so now we have 1,3-bisphosphoglycerate. You don't need to remember this name, but you can see that there's 2 phosphate groups on here. And so what that's going to mean is that it is going to rip off a phosphate group and make some ATP, and then rip off some more phosphate groups and make some more ATP. And the end game is pyruvate there. So that's the glycolysis process. Key takeaways are really just for every molecule of glucose, we are going to have 2 pyruvates that are formed and 4 ATPs that are formed, 2 ATPs that are used. So we have a net gain of 2. And then we're also going to have 2 NADHs that are formed. So in yeast, this is not something that happens in humans. Glucose, instead of turning into lactate, can change into ethanol and carbon dioxide instead. And yeast's anaerobic respiration is used in brewing, baking, and biofuels. This is called alcoholic or ethanol fermentation. And this will continue until the glucose is gone, or until the ethanol reaches some kind of toxic level, which is about 15%, which is why wine only really goes up to 15%. Beyond that, you would need to distill your alcohol. Yeast is a facultative anaerobe, which means it can use aerobic or anaerobic processes equally effectively. And NAD plus is reformed from NADH in this process of anaerobic respiration, allowing it to continue to make ATP as well as ethanol and carbon dioxide. And so obviously when we're baking bread, we know that bread fluffs, and that's because of the carbon dioxide that's being produced. Bread also actually has alcohol in it, but that just evaporates when you bake the bread. Brewing and biofuels are done a little bit differently than bread is. They are made in giant tanks, and that is going to limit the access that yeast has to oxygen. In the case of bread, it's the dough that's actually limiting the access that yeast has to oxygen. So then we're moving on to the next part. How does pyruvate move into the mitochondria and start functioning? So that we're looking here at the link reaction. And in the link reaction, we have pyruvate going into the mitochondria and being transferred to attach to a CoA enzyme group. And that's going to move it into the Krebs cycle. So firstly, pyruvate comes in through a transporter protein in the mitochondrial membrane. It's decarboxylated, meaning carbon is ripped off, and so it's going to form carbon dioxide. And hydrogen ions are removed from the pyruvate molecule during this decarboxylation process, and a NAD plus is turned into a NADH. And then it attaches to the CoA enzyme, carrier enzyme, and the 2-carbon chain, so it was a 3-carbon chain, now it's going to be a 2-carbon chain, is attached onto the CoA, and now it is called acetyl-CoA. And that's going to go to the Krebs cycle. So in the Krebs cycle, here we can see the acetyl-CoA, and it's going to join up with a molecule that constantly gets remanufactured. It's a 4-carbon molecule called oxaloacetate. So acetyl-CoA, the CoA group drops the acetyl group. So now it's a 6-carbon chain, and the CoA just moves off to pick up more acetyl groups. And that 6-carbon chain is now called citric acid or citrate. The Krebs cycle is sometimes also called the citric acid cycle. So in the process of making ATP and NADHs here, we're going to have citric acid undergoes decarboxylation, and it's going to make a NADH. And then the 5-carbon chain is going to go through decarboxylation and make a NADH. And now it's back to 4 carbons, but it's not structurally the same as oxaloacetate, so it needs to restructure a little bit. And so what is going to happen in this restructuring process is that an ATP is formed using phosphates in the matrix, and a FADH is produced, another NADH is produced, and then we're back to oxaloacetate. So for each molecule of glucose, remember each molecule of glucose makes 2 pyruvates, and as a result, this will spin 2 times. So in total, we will have 6 NADHs formed, 2 FADHs, 4 carbon dioxide molecules, and 2 ATPs. So that's the Krebs cycle. And then we are going to look at the electron transport chain. So you can see right up here, there's an image of the intermembrane space. So that's the space between the outer and the inner membrane. And on the inner membrane are all of these carrier proteins. And so that's the cytoplasm out there. This is the outer membrane of the mitochondria. And then this is the space in between. We want that space to be really tiny because we are going to have to make a concentration gradient to make most of our ATP. So firstly, NADH and FADH go to the electron transport chain. That is what this is called here in the inner membrane. And they are going to drop off their electrons, and they're going to drop off their hydrogen ions. And the electrons are going to get pumped through the actual space of the inner membrane, moving along these carriers. And the hydrogen ions are going to be pumped into the intermembrane space. And as those hydrogen ions get pumped into there, I call it a hydrogen ion dance party, but essentially you have a huge concentration of hydrogen ions forming in the intermembrane space. And the electrons flowing down the inner membrane are allowing this pumping to happen. But once they get to the end of the line, they have to be gotten rid of, the electrons. And if they don't get gotten rid of, this membrane just stops working, the electron transport chain. So then the pumping stops and we end up with way too much NADH just forming, and it's unable to drop off electrons or hydrogen ions. So NAD plus cannot reform. And then the whole ATP production process stops. So in order for those electrons to be pulled out of the electron transport chain, we need to have oxygen. And there's going to be hydrogen ions that will flow from this high concentration in the intermembrane space through ATP synthase, and that process is going to make all of our ATP. This process is called chemiosmosis. This process is called the electron transport chain, and the two of them together are called oxidative phosphorylation. So in order to get the electrons out of there, it binds with oxygen, and then also some of the hydrogen ions that are flowing back through the ATP synthase, and together they will make water. Sometimes we hear oxygen referred to as the final electron acceptor, and this is what is being talked about because they are pulling electrons out of the electron transport chain. With ATP synthase, we need to know a little bit more detail about its structure. It has only really been determined in the last 10 years. So that's what our next slide is going to be looking at here. So this purpley pink part has binding sites, and it's going to work with this brown part here, binding sites for hydrogen ions. And so this actually needs to spin all the way around. And in that spinning with the hydrogen ions in there, it is going to turn this Y section, the Y stalk is going to rotate. And that's creating the energy needed for a phosphorylation. The brown part here allows hydrogen ions to come in from the intermembrane space, and it allows them to come out in the matrix. And then we have these alpha and beta sections. The beta sections are the most important for our needs. And the beta sections allow, as the stalk rotates, the beta sections can fit phosphate groups and ADP. And as that stalk rotates, it produces the energy to join the ADP with the phosphate groups. And so there are 3 beta subunits here. in this section, and they can make 1 ATP per turn of the Y rotor. So one hydrogen ion has to pass through all 10 of these C subunits to cause one rotation. And from every 2 electrons from NADH, that's going to cause 10 hydrogen ions to be pumped into the intermembrane space. And from 2 electrons of FADH, it's going to cause 6 hydrogen ions to be pumped into the intermembrane space. So making 1 ATP is really 2.5 NADHs and 1.5 FADHs. So lastly, carbohydrates can be used to generate energy in the Krebs cycle, and they can be used aerobically or anaerobically. Glucose is a carbohydrate, and it's going to break down into pyruvate, so that's glycolysis, but then it can also go into the Krebs cycle. When we're thinking about carbohydrates, we have about 17 kilojoules per gram of carbohydrate of energy. Much of carbohydrates is oxygen, and that oxygen will not yield energy in the form of ATP. In the case of lipids, lipid breakdown happens in the cytoplasm, and it's going to make glycerol, which can go to pyruvate directly. But you'll notice it's not interacting in glycolysis, it's just making pyruvate. And then fatty acids will go directly to acetyl-CoA production. So both of these can be used in the Krebs cycle if there's oxygen, but if there is no oxygen, lipids cannot be used to generate ATP. And I've just said, so this is a typo here. Oops. Glycerol cannot be used in glycolysis because it goes straight to pyruvate here. So it's not going through the glycolysis process. Sorry about that. But lipids are going to produce about 37 kilojoules of energy per gram when oxygen is available. Most of the mass of lipids is carbon and hydrogen. So those can both be used to generate energy. Okay, so to summarize, ATP is made in small amounts without oxygen in the cytoplasm or with oxygen in the cytoplasm. Most ATP is made in the mitochondria by the electron transport chain and ATP synthase in the process of oxidative phosphorylation. The flow through ATP synthase, remember, is called chemiosmosis. But this whole process to function requires oxygen. The Krebs cycle and glycolysis are going to generate molecules of FADH and NADH, which are going to move electrons and hydrogen ions to the electron transport chain. Mitochondria have a large surface area to make this whole process happen. And if there's no oxygen, electrons can no longer move out of the electron transport chain. NADH becomes too much, and NAD+ can only be reformed in glycolysis. So we will have only a small amount of ATP produced. Fantastic. That's it for this video. Thank you for watching.